Hi, now today we are in the biology lesson. In the laboratory we have an experiment about the osmosis. We are going to investigate osmosis process about the plants. We are going to use the plants as a potato, and we slice the potatoes in the identical parts, okay? And in this process, the student Aytaj is going to help me. Before the process, we must prepare some laboratory tools. Then we will start our measurements and experiments. Okay, we are going to use concentration as a sucrose concentration in 1 mole, okay, 1 molar concentration. And we are going to use distillated water. And we will prepare 200 milliliter 1 molar sucrose solution. After that, we are going to dilute these concentrations, okay? We have different type of concentrations we will arrange on these tubes. 1 molar to 0 control concentration. Okay, now we are going to prepare the dilution or concentrations about the sucrose solution. We take 1 molar sucrose solution from the stock, and we add 4 centimeter cube distilled water into solution and add 16 centimeter cube sucrose solution, and we make the 0.8 concentration solution. And by the time in other methods, we are going to prepare the 0.6, 0.4, and 0.2 concentration solutions. And there have 0 concentration. It means that it's controlled. There is no any sucrose solution in the tube. It's just distillated water. We slice the potatoes as you see here. You can use different type of tools to slice these potatoes, okay? For example, we use the kitchen tool to slice the potatoes in identically. Or instead of these, there may be some corks or different tools you can use. Our purpose is to slice potatoes in the same sizes, and we are going to cut them, and we prepare the same length and same size potatoes, okay? After, we are going to scale these potatoes on the devices, okay? Yes, now we have the table here. We are going to write the results here, okay? And in the paper, up to down, we are going to 0, 0.2, 0.4, etc. to 1 molar concentration solutions. And we are going to indicate, we are going to write the mass of the plant tissue which we cut as potatoes here into each gaps, okay? This is the plant tissue weight before the experiment, but when we merge these plant tissues into the different concentration tubes, after 20 minutes, we will weight them, or we will scale them again and write their mass again here. And after, we will calculate the change in mass of each concentration and the percent of the change of the experiment. Okay. Yes, how we calculate the change? It's very simple formula, as you see here. We weight the potatoes after we take it from different concentration tubes. Okay, this is the change mass. Okay, we will write the change mass 20 minutes after here. But also before the experiment, we scale these tissues and we write the original mass at the formula here and multiply it with 100, it will give us the change of the weight of these tissues. At the end of the formula, we will find the change, the scale of these tissues, and what this experiment shows us in this process. It means that in high concentration solutions, we know that the plant tissues or cells will lose their water and will shrink, and actually they must, their scale must be decreased. And in control or in very low concentrations, we know that the plant tissues takes the water from outside, and their weight is going to be increased. At the end of the experiment, we are going to determine the calibration curve. Okay, we will drive a simple graphic here, and in the graphic we are going to find the isotonic point of the plant tissue. For example, in our experiment we use the potato, so this graph, this calibration graph, is going to show us the isotonic point of or concentration of the potato. It may be different in other plants. It doesn't mean that you are using the potato. Instead of potato, you can use different type of plants. And of course, in other plants, the isotonic concentration or calibration curve will be different. Okay. After this general information about this experiment, now we are starting to do the experiment with ITATs. ITATs will help us. First of all, we are going to prepare a sucrose solution. The concentration will be 1 molar, okay? I want you, ITATs, please, prepare 200 milliliter 1 molar sucrose solution, okay? There have a distillated water you can use here. Okay. Okay. You can use this spoon. Okay. We will take 6 to 8.5 gram sucrose solution and mix with 200 milliliter distillated water because of the molecular weight of the sucrose. We before calculate how to prepare the 1 molar concentration of sucrose solution. Sucrose solution is a simple disaccharide that we are using in daily. But of course, instead of sucrose solution, we may use different another type of concentration, for example, salt, etc. But in our experiment, we are using sucrose solution. 68.5 gram sucrose solution we will mix with 20 milliliter water. A little bit, please. Okay. Now take and mix it. So before you can wash inside of this petri dish, please, with distillated water into this and complete the solution to the 200 milliliter. Why 200 milliliter? Because it's enough volume for our experiment. Maybe you can use very high amounts of stock solutions, not only 200 milliliter, maybe 500 milliliter, etc. So it's enough amount we are using. Okay, now please, you are going to mix, and it must be dissolved inside of the. Okay, our stock solution is ready here, the 1 molar sucrose solution. Now we are going to dilute this stock solution and prepare different type of concentration. Okay, 1 molar is simple. We are going to use in each tube 20 milliliter or 20 centimeter cube concentration solutions. 1 molar means that just we are taking the stock solution and directly we pour this stock solution in 20 milliliter to the first concentration. To prepare the 0.8 concentration, as at the beginning of the experiment we talked about that, to prepare that, we are going to take 16 centimeter cube or cc stock solution, and we add 4 centimeter cube distillated water. It will make us the 0.8 concentration sucrose solution. And next step, we are going to take 12 centimeter cube and add 8 centimeter cube distillated water. And after 8 centimeter cube to 12 distillated water to prepare the 0.4 concentration solution. And to prepare the 0.2 concentration solution, we take 4 centimeter cube stock solution, sucrose solution, and add 16 centimeter cube distillated water to the next tube. And the last, we are not going to take any solution from the stock solution, just we add 20 centimeter cube distillated water. Okay, as we know that our solutions are ready here now, we prepare different concentration solutions here, 1 to 0.2 and 0 concentration solutions. Now we are going to prepare our materials. We are using the potato as we said before, but they may be in different sizes. Now I thought is going to cut these potatoes at the same sizes. We may use the ruler, 5 centimeter long potato is enough. And after we are going to weigh each of these potatoes, okay? When we are going to do this process, before we must write the concentrations of the solutions here. Why? Because when we weigh the potatoes, we must took the proper potato weight into its line. If we mix it, we couldn't clearly recognize that how much gram changed, okay? Just we must be careful in this stage. Okay. Now I am going to take these solutions near to the scale, because after we are scaling the weight of the potatoes, we will put each potatoes and indicate the grams correspondence to molar solutions, okay? Yes, first potato I touch now is going to scale. Yes, indicates that 2.66 grams to the 0. Now I want to show that, as you see here in the picture, now we before write the 0 to 1 molar concentration of solution and its gram mass of plant tissue before the experiment. After, we will immerse each these potatoes into the concentration. For example, this is we are going to use this potato for 0 concentration. So I put this potato near to the 0 concentration, or directly I can immerse this potato into 0 concentration. Then later we are going to weigh the second potato tissue, and we will merge it into the second concentration and we are going to write its weight, okay? Yes, now this is the last potato I thought weight at the scale here, and I put into the 1 molar solutions. After this process, these potatoes at least going to wait 20 minutes, okay? After 20 minutes, we are going to put out these potatoes and we will scale them again, okay? As you see here in the table. Again, this is the mass plant tissue after 20 minutes, and we will indicate and calculate the change in mass and percent change in mass last, then we will going to talk about what happened in this experiment. Okay, now our experiment waited 20 minutes here. After 20 minutes, we are going to take out each potatoes and we will scale them again. But I want to show you here that, as you see here, in more concentration sucrose solution, the potatoes are floating. But in 0 point at low concentrations, the potatoes are at the bottom. Then we could understand that in the osmosis processes, what is happening, the water potential or water moves from potato to outside of the solution in the high concentration. But in less concentration solutions, what is happening? It means that the potato takes the water molecules inside of its tissue and they comes to bottom of the tubes. So what this experiment shows us, we are going to find the isotonic point concentration of the potato. It looks like in the vision here 0.6 to 0.4 concentration. Between these concentrations there have isotonic value. We are going to find or estimate the isotonic concentration by this calibration curve. Yes, now it's the last stage for the experiment. Now we are going to take our potatoes. These potatoes wait in the solutions 20 minutes. ITouch is going to take out each potatoes, and ITouch will gently take its moisture, but must be careful because ITouch mustn't squeeze the potatoes because it can change our results. So slightly takes its moisture and wait, scale these potatoes again here, and we will write the mass of plant tissue after 20 minutes here in front of each concentration. Now ITouch takes the 1 molar concentration potato, so I must be careful about that. The result is written in front of the 1 molar concentration. Yes, this is the last stage. Now we are going to interpret the results about these experiments, and we calculate the change in mass and the change in percentage. And in the experiment we showed you that the isotonic concentration maybe look like different, the 0.4 to 6 concentration. Let's see the results on the graphic. This is the calibration curve graphic. I'm going to show you that where is our isotonic concentration. In high concentrations we know that there have minus change in the weight of the plant tissue in the potato because the potato loses its water. So we indicate them in minus in the graphic. But in low concentration solutions tube, the potato tissues takes the water, or in other words, the water moves inside of the tissue because they have a water potential. Then potatoes takes the or absorbs the water inside of them. But as you see here in the graphic, there have 0.4 and 0.6 between them. There have correspondent place, so it approximately is the 4.46 or near to the 4.5 is the isotonic concentration of the potato. What is the meaning of isotonic concentration? Isotonic concentration means that there is no any change. There is no any water movement inside of the potato or outside of the potato tissues or plant tissue. Okay. So this is just estimation graph because we estimated this point. We couldn't exactly say that what is the isotonic point in these experiments. Okay, thank you very much to contribute to our experimentation, and also thanks for the iTouch because she helps us in the experiments. Bye bye.